perfect. Meaning the hydrogen ions attached to something, including the carboxylic acid group. You have one on the carbon, as well as the ammonia on the other carbon. Okay? And then if there's an R group, especially if it's acidic or basic, and there are a few others that we know are titratable, those are also going to play, but at low pH, mass action favors all of those things having hydrogen. So, at very least, it's going to have a charge of +1, because in the case of the R group is non-polar and non-ionizable, the amino group will be positively charged. The carboxylic acid, although it's in its acid form, carries no charge, right? So the net charge of an amino acid that is either non-polar or polar but not ionizable will always be +1. At the other end, where it's extremely basic, you know, all those hydrogen ions that can leave to the left. And again, at the very base, so that the last thing to leave often will be it's non-polar or ionizable, will be the hydrogen now on that very weak ammonia. Okay? The hydrogen on the carboxyl group is long since gone. Okay? And this will have at best or at least a -1 charge for the carboxylic acid. Again, depending on the R group, you can vary between -1, -2. Okay? And on the other side, +1. Okay. So quickly, if we look at very simple amino acid like glycine, okay, it's going to have 2 buffer zones corresponding to the loss of the hydrogen ion, the hydrogen in the carboxylic acid group. Okay, it's then going to, once we reach that buffer zone, the pH will then increase very quickly until we get to the buffer zone of the ammonia. Okay? We have 2 buffer zones corresponding to those 2 groups. Okay? The midpoint where we have equal molar amounts of the acids and conjugate bases is known as the pK. So in this particular case, for a free amino acid, the pKa for glycine is about 2.34. And when we conjugate that to peptides and polypeptides, then this will tend to be higher. And that will tend to, the other one will tend to be lower. Okay, but for the free amino acids, these are fairly typical. Okay, so in this buffer zone at 2.34, we have equal amounts of the carboxylic acid and the carboxylate, which is the conjugate base. At the other end, at a pK2 of 9.6, we have equal molar amounts of the ammonium ion, the NH3+, okay, and the conjugate acid of that NH2. Okay. And again, the buffer zone is anywhere over this range ±1 pH. The other important point, and we're going to take advantage of this, is when is that molecule have a net charge of 0? Meaning if I put it in an electric field, it has a net charge of 0, what kind of force is it going to feel? Remember your physics. I have a net, remember Coulomb's law? Don't remind me of this cardinal. I hate physics. Oh well, too bad. We're going to use it a little bit. If I have a net charge of 0, in an electric field, how will it move? What's that? It'll move in a straight line? Well, it's exerting. Remember Coulomb's law? Remember it? No. It's the charge times the electric field is equal to the force. Right? If I have a net charge of 0, what kind of force am I going to have on the molecule? 0. So how far will it move? It won't. Right? So that's the point where I apply electric field on that molecule. We will be doing this when we talk about electrophoresis next time. Where that molecule just won't move in the presence of an electric field. And it turns out to be a very useful point. Yes?
You say to go 0.5 equivalents over, but why does it start with 0.5 and then it jumps to 1.5? Shouldn't it jump to 1?
No. Remember we talked about when we're titrating, okay? Every 1 mole of equivalent will totally titrate one of those hydrogens, right? So from 0 to 1, totally titrates that hydrogen from the carboxylic acid group. At 1/2 of that, we have equal molar amounts of COOH and COO minus. Okay, we have to go another full hydroxide equivalents of 1.5, do the same thing for the NH3 plus NH2.
Okay, and then the pKa's, those are known values, or
those are experimentally determined values. Someone actually had to sit down with a pH meter and measure. But those are, for those N groups, it doesn't matter what the amino acids, they're pretty close. And no, you do not have to know the pKs for amino acids. Yes? Right, so very simply, the way to get PI for something like glycine, which doesn't have a charged group, is you simply take the average value of the two pKs. Okay, in this particular case, 5.97. So at a pH of 5.97, glycine will have a charge of 0. Okay, yeah?
What if you have like more histidine?
Okay, so what if we have a more complex amino acid? Good question. Okay, so if I have ionizable groups, and histidine is one of them, that happens to have another pKa about 6.1. Okay, but there's others as well. Okay, so here's histidine. Okay, what you have to do is be able to relate the structures that you're seeing along that titration curve. Okay, so here we have totally titrated histidine. Okay, because we have 3 ionizable groups: the carboxylic acid group, and the NH3 group, the alpha carbonate, as well as one group on the imidazole group here for histidine. Okay, we have to add 3 hydroxyl equivalents, not 2, to totally titrate it. We're now going to have 1 extra pK. This is the pK of this particular histidine. But we need to know in order to get the PI, what are the 2 species that go into charged, okay, and that actually make it neutral. Okay? So at this point, it has a charge of -1. And at this point, its charge is 0. Okay? So here's the isoelectric one, right? So we have to take these 2, because it's this point where that net molecule has a charge of 0, right? Because it's CO minus, H3M plus, that balances out, and no net charge at that point on the other group. So you have to identify where on the curve that's going to be, and then you take the average of these 2 pKs. Okay? So you're always going to take the pKs between the 2 that give you the neutral molecule, which means you need to be able to draw all the intermediates, starting with the lowest pH and the highest pH. Okay? If there's no ionizable group, then there's just one plateau region like glycine. If there's an ionizable group, it's going to add one more region, so you'll be able to draw those structures. Okay? Okay, so again, we're always going to care about the ionizable groups on the carboxyl group and the amine group. Generally, it's about 2.2 for the carboxyl group. Strong acids, so we have 2 acidic amino acids. One of those are glutamic acid, okay, which is E, and aspartic acid, which is D. Okay, we'll have pKs that are slightly higher than that. Okay, so first the carboxyl group on the alpha carbon will go, then the R group will go, and then finally on the NH3 will go. It will be in that particular sequence. Given the pKs, you can tell what the sequence is going to be. So 3.5 to 4.5 from typical acidic amino acids like aspartic acid would be 10. Okay, the average value of pK for the free amino acids is about 9.5, and then higher for the basic amino acids. If we put them again onto a peptide or a protein that we'll talk about later, it shifts the pK up on the acidic side and down on the basic side. Again, I'll always give you the pKs. Okay, I had a couple of emails, like I said last time, you do not have to memorize pKs. Okay, you don't have to memorize pKs. I got a bunch of tests you on without having you memorize useless information that you can look up on Google. Okay? Okay, so this is just a table that shows you those typical values. Okay, here's the terminal alpha carboxylic acid group. about 3.1, what about protein? Here's aspartate and glutamate. Here's histidine. Here's the terminal alpha amino group. Okay, so it's a range. Again, these things are useful on their own sometimes because they can function as buffers, whether they're free amino acids or on protein. But again, it's the overall charge state that's going to be important. When we look at the other groups that are ionizable, cysteine, which has that free SH group, will give up proton starting about pH 7.3. pK is 8.3. Tyrosine, which is this family group with a hydroxyl group on the end, F, 10.9, so more basic than the terminal amine. Same with lysine. And then arginine, okay, because it has these resonance structures in this Biochemistry group, which I won't go into details, but you have it in your slides, it actually has a very high pK, about 12.5. That would be the last hydrogen ion in this group. Okay? So again, given the value of pK of any functional group, you can calculate the ratio of acidic and basic forms. And you had hopefully some experience. This is something you should be able to do. You did this in one of your homework questions. Okay? Which was looking at the ratio of the conjugate basis of acid and the pH unit was different by 1. And that was one question I asked you. Okay, you'll never go wrong by starting with drawing the equilibrium of that acid, writing out Ka, and then the pK, which was written again as Henderson-Hasselbalch equation, is pK equals pH plus ln the acid over the base over the acid. That's wrong, isn't it? That's wrong. That's wrong. Okay. So we could ask a couple of different things here. Okay. So for instance, for the side chain carboxyl group of glutamic acid with a P, which has a pK of 4, what is the ratio of that conjugate base to COH at 4.5? Okay. Well, I just plug in the Henderson-Hasselbalch equation. I get that the log of that ratio is equal to 0.5. That's 10 to that number, and it comes up to be 3.16 to 1. Now, I want you to be careful. These are ratios. I can ask this in a couple of different ways. I can ask, what is the ratio of the conjugate base to acid, which this would be the right answer. Or I can ask you, what percentage of that molecule is ionized? And to do that, then I have to convert this not to a ratio, but to a fraction. So for every 3.16 moles of CO minus, there's 1 mole of that. So I have to take that into account in the denominator. So if I ask you a percentage, that it's 3.16 over 3.16 plus 1, because that's the total amount. So it's about 76%. Okay, in the CO minus form, 24% as the acid. Again, the more you do problems, or the end of the book, what I will provide you with, the better off you'll be. They're not that hard, you just require practice. Okay, if you look at the NH3 group, same thing, there's the Ka. Okay, we can now do the same thing. I can ask the same question. pK of the amino group of peptide of protein is 8. What is the ratio of the conjugate base to the acid? I'll say plus 7. I plug that in. Same thing. So now I get the log is -1, which means the ratio is 0.1. And the -1 is 0.1. If I ask you again the percentage, it's 1 over 1 plus 1.1 and 91. So just be careful of what we're asking you. Okay? In the ratio, it can be a percentage. Okay, we've already talked about the isoelectric point. That is the point at which the net charge of the molecule is 0. I've given you happier with algorithms. Okay, here is the algorithm. What you have to do again is be able to draw those ionizable forms, identify where it's neutrally charged, and then take the pKs on either side, and then take the average, and that will define. Pretty simple, again, just practice is the way. Okay. So, example, okay, we look at glycine and histidine. Okay, so glycine net charge pH is 1 and plus 1. So I have to add one whole equivalent hydroxide ion to reach the pi. That equivalent point again is exactly between the two. I showed you on the graph. Those two pKs for the carboxyl group are 3, 2.34 and 9.6. That's 5.97. For the histidine, you find the two places. Okay, the equivalence point is exactly halfway between the second and third pKa. Okay, so there it's 6.0 plus 9.17, and that gives you a pi of about 7. Okay. We can then take it to the next level. Because we're going to do this. Then you had one on the quiz this week. One of the things I asked you was, what is the net charge on a peptide with how many amino acids? 4. Good. So here's an example with 4. Okay, at pH 1, okay, everything that can be protonated is protonated. Okay, in this particular molecule. Okay, so that is going to have a net charge of plus 3 in this particular example. So I have to add 3 equivalents of base to get, in this case, from the pH to the pi. If you draw out all the steps in which you can do for this with all the other groups, you'll see that's the point where that molecule, that peptide becomes 0. Okay? So you're going to titrate from the third, you're going to take the third and fourth pKs from the bottom. Those turn out to be about 6 and about 8. So the pi for that peptide is about 7. All right, so peptides are interesting. Amino acids are interesting. But what we really care about is how to use them to build peptides, polypeptides, and then the functional units that we know as proteins. That's where the real is. Emil Fisher, which you can thank Fisher diagrams for, proposed that they're made of long chains of amino acids joined by an amide bond. Now I have one chemist in here. Where's my chemist? Chemist still here? My one chemist in we already scared the chemists away. What does that tell you? They don't like biology. Or they're too afraid to identify. In organic chemistry, you probably learn about amide bonds, right? And we won't be looking at an amide bond, but in fact we have a special name for amide bonds. We're talking about polypeptide chains, which is a peptide bond. Okay? There's nothing different. It is an amide bond. Okay? So with that special name for an amide bond, we're talking specifically about joining amino acids to make peptides and polypeptides. Okay? So here's an example of a dipeptide, and when we make dipeptides, we're always going to start with the amino end and then with the carboxy end. Dr. Gu is going to want you to remember this when he talks about translation about week, 9 this course. And that'll be important there as well. Okay, we're always going to take, so if we take serine and alanine, dipeptide is called serial, we'll always put a YL between that. Starting the amino terminus, serial alanine. The peptide bond is formed by a condensation reaction, meaning we're going to have a loss of water, sometimes also known as dehydration synthesis. Okay, so every time we form a peptide bond, there'll be a loss of water. Okay, between these groups, this HN here, there's a loss of water, forming an amide bond, constraining 6 atoms to lie within a plate. Okay, and this very special structure, the peptide bond, is going to have significant sequences when we talk about higher order structures, besides just the sequence of amino acids that makes the primary sequence. Okay, then this peptide bond, okay, we have the alpha carbon, okay, which is attached to now a carbonyl, which initiated as this carboxylic acid group. The loss of water allows it now to form a bond with this amide nitrogen here. Okay, so we have 1, 2, 3, 4, 5, 6 atoms in this plane, repeating a sequence of NCC. So N and 1 N. The alpha carbon, the carbonyl carbon. Repeating again, an amide nitrogen, alpha carbon, carbonyl carbon. So it repeats. If you're a science fiction fan and you watch Star Trek, right? NCC remind you of anything? Any Star Trek fans? No Star Trek. What is wrong with this generation? No Star Trek fans? What's that? No one's admitting. That's too bad. It's our Star Wars. Here's all. NCC is the call letters for all the starships, like the Enterprise, like NCC. I can't remember not that much. But anyway, it's the repeating back from NCC, and they call it peptides. Okay? So we can have dipeptides, we can have tripeptides, and we can have obviously any number, hopefully leading to a polypeptide. And as we start to increase the number of amino acids, as you can expect, because we've got 20 amino acids, all with different chemical and physical properties, the absolute folding around all of this molecule, even though these peptide bonds are planar, is going to ultimately determine, to a very large degree, the overall folding of that molecule that we call conformation. And so we're going to spend the rest of today's lecture talking about those conformation changes. Okay? The reason why this bond is rigid or planar is because, yes? On the quiz, I was going to ask that. Oh, I'm not telling you that. You wanted to know how many amino acids on a quiz I would give. I don't give that level of detail. It's a surprise. So, planar structures, okay, are formed because of a resonance between the carbonyl and the amide bond. That now gives this some rigidity. Linus Pauling, famous chemist, was the first person to realize this. And again, Linus Pauling, as we'll find out, was also the person who identified one of the next levels of structure, which is a direct consequence of the rotation about that bond being restricted on atoms lying in the plane. Okay? So we're going to look at this sort of as a plane, because this is what we're talking about again. The peptide bond are these 6 atoms lying in the plane and constrained. So if you were to fill this, okay, so like cardboard, which people used to do with balsa wood, every time you came upon a peptide bond, you have to constrain these atoms to a plane, which greatly affects the overall foldings. Every time you come to these, you're going to have those atoms constrained. And so therefore the R groups are going to play a major role in everything else in the folding of the molecule. Okay, and again, by convention, you're going to write from the left, and the amino group on the left, ending with a free carboxyl group on the right. Okay? Again, it's an NCC repeated pattern, and between those amino acids, we're going to have peptides. Okay. So, polypeptides and proteins, we often confuse the two, although they are obviously related. Polypeptides are telling us about the polymers of the protein. A protein is a functional unit. It can be a single polypeptide chain, or it can be made up of multiple polypeptide chains. Again, you always, when we start talking about protein synthesis, polypeptide chain synthesis, we'll always start with this amino acid, methionine, okay, which is the other amino acid besides cysteine that has sulfur. Okay? In this particular case, we're tying together 4 amino acids. We will have corresponding lots of discrete water molecules, okay, forming 3 peptide bonds. In this particular case, they each constrain here. Okay? So you see the R groups hanging out here And depending on nearest neighbors, or even if it folds back, depending on the folding, we're going to start getting ionic sort of interactions, charge interactions. We have hydrogen bond interactions. We're going to have in one particular case a covalent bond, if possible. In one of those or two of those amino acids, which are identical in this case, cysteine. And we have hydrophobic interactions, but non-polar interactions. And again, when we talk about amino acids, and when we talk about how we should study the drugs, okay? If we look at an overall diagram, okay, here are obviously single letter codes of all the amino acids. Notice how this information was chunked, how it's pulled together. Okay, we have hydrophobic amino acids, we have polar amino acids. We can talk about there being small versus larger amino acids, those that have aromatic rings. And then we can sort of make this sort of complex Venn diagram to understand ultimately how they might interact with each other. Okay, to make the most energetically stable structure. So there are 4 levels. The simplest one is just the absolute sequence of the amino terminus to carboxyl terminus of the polypeptide. Okay? We have lots of possible combinations because we have 20 amino acids. And it's the absolute sequence that ultimately determines the function of those molecules. So at a very simple level, and we'll talk a lot about sequence of different macromolecules. Sequence of amino acids is critical. Sequence of carbohydrates and polysaccharides is critical. Sequence of nucleotides and nucleic acids is critical. Okay? So it's the identity of the sequence that's ultimately going to be important in defining function, or in the case of DNA coding for a protein. Okay? Again, that peptide formation is through peptide bond formation is through a dehydration synthesis between the carboxylic acid group and the amine, 2 amino acids. It's done 1 amino acid at a time, creates the peptide bond with characteristic distances between each of these, all in the order of 1.2, 1.5, 1.3 angstroms. Biochemists spend decades working out these things, but ultimately it provides valuable information for how these things are constructed. One thing you need to understand absolutely is that the sequence and the polarity is important. Polarity meaning direction, right? Whenever I talk about polarity, I'm really talking about direction. I'm not talking about electric dipoles or magnetism. I'm talking about the direction. So the absolute sequence, starting from the N-terminus, makes a difference. So this particular amino acid, let's put it in. How are these amino acids? L, leucine, glycine, threonine, valine, arginine, aspartic acid. This is the good. All you people who got it. But this has a particular secret, N to C. I can have the same arrangement of amino acids, but look, so this is not the edge of this. These are 2 completely different peptides of proteins with 2 completely different functions. They would not be identical. So it's the sequence and the polarity that's going to be critical, even though the amino acid composition is the same. When we do experiments mutating things, often what we'll do is, if we want to mutate a known sequence, we'll just scramble for the same amino acids. We shouldn't get a loss of function when we do that, because we know this to be true. But the absolute sequence, as well as the composition, is important. Okay? Sterically, because of these large R groups that are now hanging up there, the most energetically favorable orientation is going to be most of the time in the trans configuration. So in other words, around those peptide bonds, you're going to be alternating R groups. Okay, now the fold on top of that, but in general, okay, nearly all peptide bonds are going to be in the trans configuration. The other thing to notice is that even though the peptide bonds themselves are constrained in place, that we get free rotation around the carbon and the carbonyl group that can rotate. So you can think of planes of peptide bonds, but they can rotate around each other around that carbonyl curve. Okay? And we can look down from a couple of different orientations. To see that, we actually can define two different angles, okay, around this bond and around this bond. And we can define these angles as phi and psi. We can then construct energy diagram to tell us what the most favorable configurations are. And the most favorable configurations, not surprisingly, tend to favor the trans orientation. So that's all that diagram is. It's called a Rama-Shadman plot. The next level of structure is called secondary structure. Okay, so we're going to have 4 levels of structure. Secondary structure is a very specialized structure which directly relates to the fact now that we have planar peptide bonds. And we have 2 groups in that peptide bond that are capable of hydrogen bonding. We have the oxygen on that carbonyl carbon, and we have the hydrogen on that amide nitrogen. Both of those are electronegative groups. Both are capable of hydrogen bonding. But the problem is it's not going to happen when the distance is too great. You will remember that hydrogen bonds operate over relatively short distance, of the order of 1, 1.5, 3 inches. The distance between adjacent amino acids is greater. So in order to have these types of interactions between these groups, some longer distance has to occur. And out of this, we get some very special structures. One of these is known as the alpha helix. Okay? And in the old days, I can still do it. They keep this around for me, because none of you guys know what these things are anymore.
What is that thing?
Yeah, that's what I mean. This is an alpha helix. Okay? These old phone cords are actually good examples of it. I hate wireless. So I lose my demos. Okay? So this is sort of what an alpha helix can look like. There's all helices, there's a characteristic repeat distance. Okay, and that repeat distance, as I'll show you in a minute, has to do with the repeat distance between peptide bonds when a carbonyl oxygen binds down from a few amino acids to the hydrogen on the amide. The other repeating structure is called a beta-pleated sheet, where we have a stretch of amino acids in the peptide that backs on itself, okay, and then comes back and then interacts amide, hydrogen, carbonyl, all on a stretch. And that causes that stretch of protein to buckle to form a pleat, known as a beta-pleated sheet. Okay, so that's a highly ordered structure which are very specialized. Okay, if we look at these, okay, so for instance, if we look at either one of these, with an alpha helix, we have to go 1, 2, 3, and a little bit, 3.4 roughly, amino acids. So that this carbonyl oxygen interacts with this hydrogen on this amide. Okay? If it will be the next group, so it will be this one and this one. Okay? This is going to lead to this spiral known as the alpha helix. So whether we're talking about an alpha helix or beta helix, we're looking at highly ordered structures due to interactions, hydrogen bonds between those 2 atoms in the oxygen, the carbonyl oxygen, and the hydrogen on the amide. Right? Okay, it's just hydrogen bonding, but many of these hydrogen bonds can be quite strong. Okay, so think of Velcro, right? Have you all looked at Velcro on your microscope? Yeah, y'all look at Velcro on the microscope. You know what Velcro is made up of? You should look at this. It's a brilliant structure. One side, the soft side of the Velcro, is nothing more than a fabric with lots of loops on it. The other side is a plastic that has a hook on it, right? That's what Velcro is. One of those is rather weak, right? But as you start to add hooks and loops, it gets stronger and stronger. Right? A multiplicative action. Same with hydrogen bonds in a structure like an alpha helix. An alpha helix that has a number of these bonds can be quite strong. It's not the first time we'll see strong structures that result from hydrogen bonding. A DNA double helix is another example of that. Many, many hydrogen bonds make that duplex structure very strong. Okay? So, because of that, we see a lot of alpha helices in what we call structural proteins. Okay? The alpha helix, also the beta conformation. You see various types of proteins that convert back and give them special names. There's their unusual structure. Collagen is actually a helix, okay? And then it's a helix within a helix. So a single polypeptide in collagen is an alpha helix, and then there's 3 of those that get bound together to make the collagen molecule itself. So these are very important for structural integrity. So this is always what's called a right-handed structure, which means if I look down up on the bottom, it will coil this way, okay? Which I used to call clockwise, but I now know that none of you know what a clockwise is. So it's a digital format. Okay, there is a right-handed, okay, or right-handed screw. There's about 3.6 amino acids for every 360 turns. The repeat distance, known as the pitch, is about 5.4 angstroms. Again, the peptide bonds are planar, and the bonds between the carbonyl, oxygen, and the hydrogen are. And all the R groups face outwards from the helix. So you think of it almost as a spiral staircase with the R groups sticking outside the helix. Another class of molecules that we see a lot of alpha helix is, especially when we use hydrophobic, non-polar amino acids, is when we build channels and membranes. Those channels tend to have a lot of alpha helical nature with those non-polar R groups, which allow it to go through those amphipathic phospholipids through those helical chains. Okay, in simpler forms, okay, it looks like this. Okay, if you look down through the center, you will see that there's a 4 with the R groups hanging out. So very elegant structures. Linus Pauling was the one who came up with the alpha helix, one of his 2 Nobel Prizes. Okay. So several factors can disrupt alpha helix. Okay. Proteins, because of their bizarre nature, not being an amino acid but an amino acid, as I told you last time, whenever you see a protein, you will often introduce a kink, because that NH group now is tied up in that cyclic structure. And that introduces kinks in all kinds of molecules. So that leads to restricted rotation around that structure. Strong electrostatic repulsion caused by proximity of acidic side chains or basic side chains. Right? So if they're close to one another, the light charge, the electrostatic will repel. That will disrupt those helices, overwhelm the hydrogen bonds. And then steric hindrance caused by very bulky side chains like daily isolation and creatine can also disrupt. So that's just to say that the nature of the R groups is important in the actual integrity of all conformation, but in particular secondary structure. Okay. The beta conformation of beta pleated sheet uses again the same groups, those oxygens on the carbonyls and hydrogens on the amides, but forms creases, and the alpha carbon are the creases themselves, the peaks and troughs of those pleats. But again, it is stabilized by those particular hydrogen bonds between carbonyl, oxygen, and hydrogens on the okay? And they're perpendicular to the direction of the back. So here's some examples. They can either be anti-parallel, meaning you'll have from the amino end this way, run along a stretch, make a U-turn, and come back. That's anti-parallel. Okay, so at the end of the chain, at the end of the sheet, you can see an N and a C. On the other hand, they can also be parallel, comes along, right? Does a little something up here, and then comes back along again in the same direction. That's just a parallel sheet. Believe it or not, people get paid to figure out these things. But they are important. Okay, we have all sorts of weird things. We've got beta turns, sometimes called reverse turns, which is just a abrupt change in the direction of the polypeptide. Often these are caused again by introducing amino acids like proline, or very small amino acids like glycine, asparaginine, and serine, which are small hydrophilic residues. All of these things introduce a rapid turn. And beta sheets are found in a lot of interesting proteins. So, for instance, a fatty acid binding protein, something that binds fatty acids, has a barrel-like structure with a lot of anti-parallel beta-free sheets that these arrows represent. So here's N to C, N to C, N to C. You'll see there's the wings in an anti-parallel fashion, forming a barrel-like structure. Another famous molecule these days is green fluorescent protein. Okay, it's actually from a jellyfish. It binds calcium. It can actually be fused with genes, including genes from humans. And so you can actually follow their translation, their gene expression, because this is naturally fluorescent. You can follow it in a microscope to see if that particular gene is being expressed. We have a number of super secondary structures. I won't spend a lot of time, in fact, hardly any on these, but we have various different kinds of combinations. It's not that important for this course, but you should realize that you can get all sorts of combinations of these types of compounds. Okay? So again, there are some more barrels and more stuff. The next level of structure is called tertiary structure. It's very simple. Once we get beyond secondary, we're a single polypeptide chain. The overall folding of that protein, which is governed primarily by the R groups now, is going to be in an energetically favorable state, lowest free energy possible. Okay? The absolute sequence of amino acids is important. The solvent that it's in is important. The temperature, the ionic strength, all the environmental factors are important to lead to an overall folding. All of those interactions together are known as the tertiary structure, but it's for a single polypeptide chain. There is one additional bond here, which is because of the amino acid cysteine, which can be oxidized to make cysteine or disulfide bridge. It's the only covalent bond between amino acids other than the peptide bonds. Okay, so within tertiary structures, we'll often see disulfide bonds. Interestingly enough, if you remember your cell biology, you know that translation of genes or mRNA proteins can occur either in a cytosol, where polypeptides and proteins remain there, or they can occur in the ER. Those proteins are either secreted or end up in membrane proteins, or end up in the lysosome. In those particular cases, the ER itself is an oxidizing compartment. So only proteins that go through the endoplasmic reticulum, go through the endomembrane system, will have disulfide bonds. And they are very strong because they are covalent. In order to break those bonds, you need to reduce it. Reduce in the lab with reducing agent. Otherwise, that is a strong covalent bond. So, for instance, insulin is an example of a molecule that actually is expressed as a single polypeptide chain initially. There are what's called post-translational modifications that cleave portions of that molecule off. So it looks like a more complex molecule, but it's really only one gene product. Is those two chains are stabilized by disulfide bonds within the A chain in one place, between those two cysteines up there, and then between these two chains, okay, after it's processed. So it looks like there were two polypeptide chains, and strictly speaking, that's what the process looks like. So it starts off at the N-terminus, you make the entire protein that's called pre-proinsulin. Okay, there's a sequence that sends it to the ER called the signal sequence. That gets cleaved off. That gives us what's known as proinsulin. And then an enzyme known as a protease comes in and cuts off one of those chains, chain C, which gives us two apparent polypeptides. And it starts off as a single polypeptide. Okay? But again, we're going to see multiple molecules which can be characterized by that. Finally, we talk about quaternary structure. The interactions in quaternary structure are identical to tertiary structure, except there's one key difference. There's more than one polypeptide chain. Okay, so all the interactions I just spoke about, all the weak interactions, as well as the covalent interaction with cysteine, it is responsible for establishing quaternary structure. But in this particular case, to make it a functional protein, it needs to have more than one polypeptide chain. And usually that is because it is 4 different gene products. Okay? The classic example of quaternary structure is hemoglobin. It is made up of 4 different polypeptide chains, known as subunits. 2 are known as alpha subunits, 2 are known as beta subunits. They will come together to work as a functional unit. Okay? Histidine plays a major role in establishing a unit called a porphyrin ring, which coordinates up to 4 oxygen binding sites on this molecule. It shows some really extensive behavior that we'll talk about on Friday called cooperativity to give us this functioning program. Okay? On Wednesday, we have the next lecture.